Hey friends, welcome back again to LCG today with a new video, and today my topic is regarding biochemistry, and it is part 2 of biochemistry. So in this we will get discuss about the Krebs cycle. So basically Krebs cycle is also known as the tricarboxylic acid cycle or citric acid cycle. Basically this cycle is completed inside the 8 steps. Fine, so this is the cycle, and here is the trick to learn the Krebs cycle. So it is a one word sentence that's easily you will recognize this cycle in easy manner. So this is the trick to learn Krebs cycle here. Can I keep selling sweets for money, officer? So here is the C for citric, I for isocitrate, K for alpha ketoglutarate, S for succinyl coenzyme A, and S for succinate. So this is the whole trick to learn this. After that we will move to the next slide. Here F for fumarate, M for malate, F for fumarate, M for malate, and O for oxaloacetate. So basically it is a 8-stepped chain reaction, and it is a cyclic pathway where it composed of the main series. Like here it is the pathway. Here you can see it is in cyclic form. So we will discuss the enzymes which are given in this yellow colored reaction. So this is the trick to learn them in an easy way. So what you will need to do is you will have to learn this whole cycle. So Krebs cycle is basically what we will complete this in a basic manner. That Krebs cycle has 3 names of itself. It is also known as the tricarboxylic acid cycle plus Krebs cycle plus citric acid cycle, and it is a series of enzymes which are completed in 8-stepped reactions. It was first named by Hans Krebs, and he postulated the detailed cycle. For this thing he also given award as a Nobel Prize in 1953 for his contribution itself. So Krebs cycle basically it takes place in the mitochondrial matrix. Either it is in the inner matrix or inside the outer matrix, where acetyl coenzyme A getting oxidized and it forms carbon dioxide, as well as coenzymes getting reduced, which generate ATP in the electron transport chain. Here you can see there are some ATPs getting produced in the manneric form. You will see here, right there you can see in this step, here is GTP, ATP and ADP. So these are the steps which produces ATP. What does ATP mean? ATP means the energy. Energy is the higher sign. So we can basically use this simple arrow form. This is the basic arrow which means energy. Energy, which means having to increase something in the cyclic pathway, like ATP we're talking about the GTP, we're talking about the ADP. So all of these things getting used, like ATP, adenosine triphosphate, I will talking about ADP, adenosine diphosphate we are talking about. So these are basically what? These are energies, and to complete this whole cycle we need to do what is the energy released, and these are yellow colored. These are the enzymes. These are the enzymes like isocitrate, alpha ketoglutarate, succinyl coenzyme A, maltase, fumarate, and citrate, oxaloacetate and succinate. So these are basically what? These are the cyclic pathway enzymes to complete the Krebs cycle in the whole cyclic pathway. So next we will move to the here is the trick that I told you previous, that it is a one-line trick for the easy learning trick to learn the Krebs cycle. So these are basically the enzymes which are getting used in this pathway. So here are some steps of Krebs cycle. So the first step of Krebs cycle is that I told you it is a 8-step process. To complete the whole cycle, we need to do what is it is completed in the 8 steps, and these steps of this cycle takes place in the matrix of mitochondria under the aerobic conditions. In first step, the condensation of acetyl coenzyme A with 4-carbon compound oxaloacetate to form the 6-citrate coenzyme A is released, and the reaction is catalyzed by which enzyme, that is citrate synthetase. If we are talking about the second step, citrate is converted into its isomer. What is the isomer of citrate? That is isocitrate with the help of enzyme actonitase. Fine. So here is the cyclic step that I told you in a previous manner, that what these are the, this is the enzyme, citrate synthesis, this is the enzyme, oxaloacetate, this is the enzyme, isocitrate, this is the coenzyme. So basically what you have to do, you have to learn that it is a 8-step process. It takes place in the matrix of mitochondria under aerobic condition, and first step is converted is the condensation of acetyl coenzyme A with 4-carbon compound oxaloacetate to form 6-citrate coenzyme A is getting released, means it is released. Next, citrate is converted into what? Isocitrate with the help of enzyme actonitase. So it's all about the Krebs cycles. These are having some basic steps. This is the first step, this is the second step. So you can clearly recognize actonase, citrate synthetase, oxaloacetate and isocitrate. And it is a 8-step process, takes place in the matrix of mitochondria under aerobic condition. So these are the first 2 steps of the Krebs cycle, and you will easily recognize these steps. Further you can see back here, you can see the cyclic pathway that we are talking about. This first step, citrate, and this isocitrate. So we are discussing the step 1, and this is the step 2. So we will what discussed is step 1 and step 2. For step 1, citrate is required. For step 2, isocitrate is required. So in next one, we will talking about the steps like alpha ketoglutarate, succinyl coenzyme A, succinate, fumarate, maltase, oxaloacetate, and so on. And we are talking about these ATP releasing compounds. So let's begin with the next slide. Okay, so 2 steps we will discuss. In step 3, what does it do? Isocitrate undergoes dehydrogenation. When here is dehydrogenation of something, it means it is the removal of watery substance. Okay, so here it undergo dehydrogenation and decarboxylation to form what? 5C alpha ketoglutarate. And what enzymes it is dependent on enzyme NAD positive, and NAD positive is getting converted into NADH. In 4th step, alpha ketoglutarate undergoes oxidative decarboxylation to form what? Succinyl coenzyme A with 4 carbon compounds. Alpha ketoglutarate is released what? 1 molecule of carbon dioxide and NAD positive, and NAD positive again converted into what? NADH. So here NADH is converted. Why so? Because NAD positive is getting converted into NADH. This NADP converts what? NADH. So NAD positive is an important compound because it produces NADH. Fine. So this is all about the pathway of this reaction. So it undergoes what? Dehydrogenation, decarboxylation to form what? Alpha ketoglutarate, and produce what? Dependent enzyme is what? NAD positive, and NAD positive will convert NAD into NADH. Same as the case is here, only the alpha ketoglutarate produces dehydrogenase enzyme complex and releases carbon dioxide, that is CO2, with the help of NAD positive. This is the third and fourth step. In next step, in fifth step, succinyl coenzyme A forms what? Succinate enzyme with the help of enzyme succinated into FADH2 combined, and what it form is NADP positive forming. If and you will know previous that NAD positive will releases what? NADH enzyme. So malate here is dehydrogenase catalyzed reaction. As you know earlier that NAD positive will forming what? NADH. So malate dehydrogenase catalyzes this reaction. Mean malate dehydrogenase is important in step number 8. And in step number 7, here furanase is important. Here is what? Malate plus water is added in 7th step, and in 8th step malate dehydrogenase and catalyzes this reaction overall. So you can again see the cycle in next step. Back we will getting into the slide. So in this cyclic pathway you can see that all the steps we are getting discussed. These are the cyclic pathway, and these are the enzymes which are getting converted from 1 to 8 step. So these are the enzymes which are helpful for producing NAD and NADH, NADP positive, and ATP to convert is what? ADP, that is adenosine triphosphate. So ATP, that is the energy currency of cell, because energy is most important step for this cyclic pathway. If energy is not there, it will never getting converted into the whole pathway. So it will increases the energy, and energy is very important for this whole cyclic pathway to complete it in the monadic and well maintained form. So that's all about the Krebs cycle today. I think most of the things getting clear, that it is a 8 cell cyclic step and it is very easy to learn with the one trick of pathway. So thank you friends, thanks for watching this video, and if you get something from this video, so I hope you will get some informative information regarding biochemistry. So thank you friends, keep subscribing and like, share. Thank you.